Genetically engineered organisms are also referred to as bioengineered or genetically modified. The rhetoric surrounding the issue changes depending on who you talk to, but the implications do not. This project studied the presumed health effects of the genetically engineered food that we consume on a daily basis. What is it? Genetically engineering an organism is the changing of a genome, inserting new DNA into an organism so that it is replicated and becomes part of that organism's inherited line of traits. According to the Center for Food Safety, 85% of U.S. corn is genetically engineered, as are 91% of soybeans, 88% of cotton. Cottonseed is used in a lot of foods. According to industry, up to 95% of sugar beets are now genetically engineered, and it has been estimated that upwards of 70% of processed foods on the supermarket shelves, from soda to soup, contain genetically engineered ingredients. How it's done. Genetically engineering an organism is done by manipulating plasmids. Plasmids are rings of DNA in bacteria. They are cut using specific enzymes and somehow manipulated by scientists. Scientists can remove a segment of DNA, add a segment, or replace a segment with a different gene. Once the plasmid has been altered, it is referred to as a vector, which is a channel used to carry DNA into the host. Once the plasmid is changed, it is reinserted into the host, and now the host genome contains a new gene. This is an example of a plasmid. The gray area represents the gene that is being replaced. Each nucleotide up top codes for a specific amino acid. So the gray area will be swapped with a different series of nucleotides. Genetically engineered foods are controversial in society because of presumed health risks and the implication for private agriculture. Both of these issues will be discussed in this project. Recently, California's Proposition 37 regarding the labeling of GMOs has brought the issue to the table for a lot of citizens. The current policy. The FDA regulations currently state that the law requires all food labels include a name that accurately describes the basic nature of the food. Regarding bioengineered foods, the name changes are only appropriate when the food is significantly different from its traditional counterpart, such that the common or usual name no longer adequately describes the new food. Significant differences in food arising from production processes must be disclosed in labeling. So if a transgenic production method materially changes a food's nutritional profile or results in safety concern, this must be disclosed on the label. The definition of material difference is not actually specified, but precedent guides the FDA in its interpretation of the term. The FDA allows voluntary labeling as long as labeling is not false or misleading. Further in depth, the AMA policy points are as follows. There is no evidence that unique hazards exist either in the use of DNA techniques or in the movement of genes between unrelated organisms. The risks associated with the introduction of DNA-engineered organisms are the same kind as those associated with the introduction of unmodified organisms and organisms modified by other methods. Assessment of the risk of introducing DNA-engineered organisms into the environment should be based on the nature of the organism and the environment into which it is introduced. So this is from the American Medical Association, and the bottom line is that every case is unique. To me, the key phrases in this are material difference, which is not defined and is left to the discretion of the FDA, so we're not quite sure if we agree with them or if they are making our decisions for us. And also, they urge consumer education is completely necessary, and it seems to be missing from a lot of people's curriculum. So here's some consumer education. If we use corn as the example food, the question is, why would we want to change something found in nature? If you inject the DNA of a bacteria that produces a Bt toxin, the corn will replicate that DNA and you get corn that is resistant to insects because it contains Bt toxins, which repel insects. Now, that could be a good thing if you're a farmer, or you could look at this and feel very uncomfortable about eating something that was just essentially injected with toxins. You would be right to be inherently uncomfortable. Once we ingest the corn, the gene that produced the Bt toxin is in us as well. If the gene that creates the Bt protein were to transfer to our intestinal bacterial cells, our intestines would become a veritable host to a factory of pesticide manufacturing. DNA has been shown to transfer from our food to any organ in our body, even fetuses. It may seem highly fantastical that DNA would transfer cells, but to put it in familiar context, viruses are DNA with a protein coat. If you get a virus by inhaling someone else's sneeze, doesn't it seem reasonable that you can also get a virus by ingesting something? Germs are not a foreign concept to us. We are just unused to thinking of our food's genes as germs. More health effects. According to the Center for Food Safety, some generic ones are toxicity, allergenicity, antibiotics resistance, immunosuppression, and cancer. The environmental impacts would be the use of the genetic engineering in agriculture, leading to uncontrolled biological pollution, threatening numerous microbial plants and animal species, and the potential contamination of all non-genetically engineered life forms with novel and possibly hazardous genetic material. The Center for Disease Control received an adverse event report from the FDA due to people complaining of health issues from eating Starlink corn products. The CDC conducted a survey of the affected people and issued a report to the FDA about the harmfulness of foods with modified genomes. The CDC collected serum samples from the individuals that ingested the Starlink corn and tested them for evidence of allergenic responses to the Cry9C protein. Further studies by the American Academy of Environmental Medicine have proven that Bt crops have been linked to sterility and disease. There are just a few statistics from animal studies concerning genetically modified soy. Over 50% of the offspring of a mother rat fed genetically modified soy died within 3 weeks. Female rats fed genetically modified soy showed changes in their ovaries and uterus. By the third generation, most hamsters that were fed genetically modified food were unable to have offspring. In addition, rats that were fed genetically modified potatoes had excessive cell growth, which can be seen in their stomach lining and intestinal walls, which is reminiscent of cancer growth. A case study about the health of genetically modified stuff would be L-tryptophan, which it may sound familiar because it is the stuff found in turkey that makes you happy and sleepy. Tryptophan is a necessary and naturally occurring amino acid that is a precursor for serotonin biosynthesis. Serotonin is a chemical you secrete that makes you happy. So tryptophan can be manufactured and prescribed as medicine for many conditions like depression and insomnia. In the 1980s, there was an epidemic of eosinophilia myalgia. The symptoms of this disease are severe myalgia, which is muscle pain, increased white blood cell count, joint pain, cough, fever, and this was caused by the ingestion of tryptophan, but only from one manufacturing site, which was in Showa Denko in Tokyo. The production of tryptophan involves bacteria fermentation process in which multiple secondary metabolites are involved, and there's a long purification process. Analysis of the manufacturing procedures at Showa Denko revealed several simultaneous changes in manufacturing protocols. These included the introduction of a new bacteria strain that had been genetically altered to increase the production of L-tryptophan. This is the fourth largest chemical company in Japan, and they were able to change numerous protocols in their production of L-tryptophan. And in addition, this product exceeded the FDA standard for pharmaceutical grade by more than a full percentage, which is disturbing because it begs the questioning of the logic of the system. Many people have recently become aware of the Monsanto problem, which has received controversial attention. Monsanto is a company that sells and trades seeds developed through biotechnology and works with crop protection through chemicals. The Agricultural Appropriations Bill has an attached section called the Farmers Assurance Provision, which was attached to the Agricultural Appropriations by the subcommittee chairman, Representative Kingston, under pressure from Monsanto. The rider bill is, quote, nothing more than a sneak attack on American farmers, consumers, and the environment, according to the Organic Consumer Association. If the bill was passed, Monsanto would have immunity from federal law by allowing the biotech industry to plant genetically modified crops. Federal courts have acknowledged that the inevitable genetic contamination poses a threat to a farmer's choice to grow non-genetically engineered crops and a consumer's choice to eat them. The Monsanto Protection Act would make judicial action to protect farmers and consumers toothless. Here are some of my sources. And to go back to the bottom line of this presentation, when the genes are not selected by nature, I propose they are not stable. There has been things introduced by man to the environment before, like the grapefruit, and geneticists acknowledge this, but we've never been manipulating a genome before, and there's cause to worry about the possible consequences of having a gene that looks for its place